Hello, it's Sally again. Welcome to Biosignaling 3. This is the last lecture on biosignaling. And, in this lecture, in the last lecture, I'm going to talk about, some of those concepts that I didn't talk about in previous lectures, including, nuclear receptors. I talked about mostly those membrane-bound receptors. I didn't talk about nuclear receptors. So I'm going to talk about their structures and how they function. And I'm also going to talk about and introduce a very important signaling molecule, which is, or a signaling protein, which is calmodulin. It's very common and very in many biosignaling pathways, and it's a very important molecule. And finally, I'm going to introduce CaM-kinase enzymes, or those calcium calmodulin dependent kinase enzymes, particularly the second type, type 2. So this is just the outline of Biosignaling 3. So let's begin. So we had the same figure in, I think, Biosignaling 1.1. I talked about most of these. I talked about most of these receptors. I talked about ligand-gated ion receptors, ion channels, actually, G-protein coupled receptors, those enzyme coupled receptors. But as you see, all of them are membrane-bound. All of them are embedded in the lipid bilayer of the membrane. But we have another type of receptor, these nuclear receptors, which are in the nucleus, located in the nucleus. And as you remember, we have two general types of signal molecules. Those which are very large or hydrophilic, so that they cannot pass through the lipid bilayer. So they need to be received at the surface of the cell, and they are actually received by these cell surface receptors, okay, which are embedded in the membrane. But there are some small hydrophobic signal molecules which can actually, because they are small and hydrophobic, they can pass through the lipid bilayer of the membrane, and then they are carried to their receptors by some specific proteins, and they are received in the nucleus. Basically, there are those signal molecules which activate the transcription of a gene. Okay, so they need to be received and be processed in the nucleus of the cell. Okay, so now let's talk about the general structure of a nuclear receptor. This is the general structure of a nuclear receptor in the inactive state, of course. Excuse me. So we have three main domains. We have a DNA binding domain, which binds to DNA, of course. We have a transcription activating domain, which binds to transcription factors. Transcription factors, or co-activators or co-repressors of transcription factors. And we have a ligand binding domain. And as you see in the inactive state, we have an inhibitory protein which inhibits this ligand binding domain. But what happens when the ligand binds is that this inhibitory protein is detached. So the ligand binding domain here is actually clamps shut, okay? And the whole receptor undergoes a conformational change. The DNA binding domain binds to a specific, portion of the DNA or gene, and this transcription activating domain binds to a co-activator protein. And once this co-activator protein binds, it may recruit some transcription factors, which then activate the transcription of a specific gene. In another scenario, which is not shown here, instead of this co-activator protein, some co-repressor proteins may bind, and they actually inhibit the transcription of a gene. So, yeah, it's not shown here, but it's another case. So this is the general structure. But a real example is this ribbon structure of the retinoic acid receptor. And as you see, it is inactive right now. But when the ligand binds to it, it undergoes a conformational change. And you can see that conformational change mostly at the bottom of this receptor. And what happens next is that thanks to this conformational change, as a result of the ligand binding, the alpha helix of a co-activator protein can then attach to this part of the receptor and, as I told you, recruit some transcription factors and activate the transcription of a gene. So this is a real example of what I explained here. So that was it. That was all I wanted to talk about nuclear receptors. There are a lot, of course, but this is just, I wanted to just give an introduction about them. Yeah. Okay. Now let's talk about calcium calmodulin. It's a very important molecule. Well, calcium calmodulin, the reason it is named calmodulin is because it is modulated by calcium, okay? It's actually a sensor for calcium. It senses the concentration of calcium in the cell, and it is activated by calcium ions, okay? So when it is not bound to calcium, or when calcium ions are not bound to it, it is in the apo form or the inactive form. But once the calcium ions bind to that calmodulin, it undergoes a conformational change again, and it is now in the active state or halo form. So we have two. So it can be in two states: apo or inactive state, halo or active form or state. And this is, as you see. Okay, fine. This is, calcium calmodulin has a dumbbell shape, and there are two globular ends, okay? And each one of these globular ends has two calcium ion binding sites, okay? So two in this end, two in this end. And there is a central tether region, or the central alpha helix. And this alpha helix is very important. It's because it is very flexible, and it allows the calcium calmodulin to take different conformations or take different shapes, okay? And bind to a lot of different target proteins, almost up to 100 target proteins, okay? So, as I've told you, these calcium ions bind to these globular ends, and now, this is the halo form, actually. This is the active form of the calcium calmodulin, and what happens next is that it can wrap itself around a peptide portion of a target protein, okay? And by doing this, it actually activates a target protein. And I'm going to explain, for example, a real example in which calcium calmodulin activates a kinase enzyme, okay? So this is the general structure of calcium calmodulin. So this is the apo form that I talked about. As you see, the main feature of the apo form, I guess, is in this part. This central tether region, or this central alpha helix, is not straight. But once the calcium ions bind, it is a strength. It's now straight, and it is in the halo or active form. Okay, this is from another view, and these, this one and this one, these two are those two calcium ions which bind to one of those globular ends of the calmodulin. So, it's a very important molecule, actually, and this is this structure. Fine. Now let's talk about the calcium calmodulin dependent kinase II, or CaM-kinase II. Well, this is a kinase which is activated, or its activation depends on calcium calmodulin. This is the line diagram of its gene. There's a catalytic site, there's a self-association site, there's an autoinhibitory site. These two phosphorylation sites are really important. I'm going to actually explain why. This picture you see, this is a computer-generated picture of CaM-kinase II based on the data from high-resolution electron microscopy and X-ray diffraction techniques. I know something about electron microscopy, but I don't know a lot about X-ray diffraction techniques. The only thing I know is that scientists make a crystal of a specific protein, and then they irradiate that with X-ray, and they just try to understand where are the electron densities and some stuff like that, and they figure out the structure of a protein. Okay, so this is basically the picture generated with those techniques. So what you see here is that this is the picture from the top. 1, 2, 3, 4, 5, 6. There are 6 kinase enzymes. Okay, these are kinase domains. They are bound together. They are held together by 6 Haupt domains. Okay, there are 6 Haupt domains. Sorry, and 6 kinase enzymes. And but this is not the real structure. This is the picture in Figure C. This is the stereo rendering of the same structure here, but it actually shows 2 of these rings. So we have a ring, but in the real CaM-kinase II, we have 2 of these rings. So, we have in a ring, we have 6 kinase enzymes gathered together by Haupt domains. Okay, so we have a ring, but in real calcium channel modulating dependent kinase II, we have 2 of these rings stacked together. Okay, so in the real enzyme, we have 12 of those kinase enzymes. Okay, and in Figure B, you see only 1 of those rings. But in Figure C, this is the side view of the same structure, and you can see this is the top view, but this is the side view. And when you look at it from the side, you can see that there are 2 of these rings, and in general, 12 kinase enzymes. Okay. So calcium channel modulating dependent kinase II is specifically enriched in the nervous system, and it's one of those molecules which has some important roles in the induction and expression of long-term potentiation. I'm actually going to explain that later in the lecture on explicit memory, but it has a lot of important roles in signal transduction pathways in the nervous system. Okay. So now let's see how calcium channel modulating dependent kinase II, or CaM-kinase II, is activated. So this is the CaM-kinase II in the inactive state. Actually, when CaM-kinase II is in the inactive state, it is in a dynamic equilibrium between 2 states. In one of those states or forms, let me actually explain these domains first. We have the kinase domains. Okay, these 6 kinase domains. This is one of those rings, as I told you. In a real CaM-kinase II, we have 2 of these rings stacked together. But this is here we just picture one of those rings. So in one of those rings, we have 6 of these kinase domains. We have again six of these Haupt domains, and they're basically gathered together by these linkers, and each kinase domain has its regulatory segment and phosphorylation site. Okay, so when CaM-kinase II, as I told you, is in the inactive state, it is at the equilibrium. It is at the equilibrium between 2 states. Sorry, I forgot what I wanted to say. So it is at equilibrium between two forms. In one of these forms, all of these kinases are stacked together like this, but sometimes, the CaM-kinase is still inactive, but sometimes one of these kinase enzymes pops out. In this form, the regulatory segment of these kinase enzymes is buried in the kinase active site. But when one of these kinase enzymes pops out, the regulatory segment is now, can be accessed by calcium channel modulating. So when the calcium calmodulin complex is present in the cell, the CaM-kinase, calcium calmodulin complex can, binds to this regulatory segment of CaM-kinase II. Okay, and now it is in the active state, because its regulatory segment of this kinase enzyme is now inhibited by CaM-kinase II. Calcium calmodulin, and now the kinase is active. So again, when it is inactive, it is at a dynamic equilibrium between these two states. The regulatory segment sometimes is buried in the kinase active site, but sometimes one of these kinase enzymes actually pops out, and when it pops out, its regulatory segment is available for the calcium calmodulin. So whenever the calcium calmodulin is present in the cell, the calcium calmodulin complex is going to bind to this regulatory segment and activate one of these kinase enzymes. And now it is active. What happens next is that another one of these enzymes may pop out again, okay, and again calcium calmodulin, calcium calmodulin complex may activate that. But a very important phenomenon, something very important may happen, and that is autophosphorylation. Sometimes, there is a phosphorylation site for each one of these kinase enzymes. And sometimes these kinase enzymes are autophosphorylated. This autophosphorylation has a very important role. It actually prolongs the active state of CaM-kinase II. And the way it does that is first by trapping the calcium calmodulin in place. So it traps the calcium calmodulin in place, and by doing this, it prolongs the active state of CaM-kinase II. And this calcium calmodulin stays there until the intercellular concentrations of calcium is decreased. Okay. So it stays there, and even if after the detachment of this calcium calmodulin complex, this kinase enzyme can still be active thanks to this autophosphorylation. Why? Because this autophosphorylation, the second way it actually prolongs the active state, is by making this CaM-kinase II a calcium-independent enzyme. Okay, because even after the detachment of calmodulin, it can be still 50 to 80% active, and it is it turns into a calcium-independent enzyme. Okay, and it stays active until a protein phosphatase dephosphorates these two phosphorylated sites, and a protein phosphatase actually turns it into an inactive enzyme again. So this autophosphorylation is very important. It prolongs the activation or the active state of the CaM-kinase II by trapping the calcium calmodulin in place, and by turning the CaM-kinase II into a calcium-independent enzyme. So even after the detachment of calcium calmodulin, it stays active until a protein phosphatase dephosphorates that. Okay, and this is just a spectacular quaternary structure in ribbon form of calcium calmodulin. It's very beautiful. I'm fascinated by that. So that was it. That was all I wanted to talk about. CaM-kinase II. That's done. It's finished. Okay, so the whole focus of these lectures on biosignaling, was that the main purpose was that I wanted to review some of the basic principles of biosignaling and signal transduction pathways, so that I've reviewed and covered the those basic concepts that I need for explaining some other concepts in future videos. Because I'm going to actually explain, as I told you, I'm going to explain explicit and implicit memories, their molecular and cellular aspects, and I needed some of these concepts to fully understand the molecular aspects and molecular neurobiology of explicit and implicit memories. So that's it. Thank you so much for watching these videos. I hope you enjoyed them. And yeah, have a nice day.